Artificial Intelligence Doctor. User question. Can dietary components switch off PSKH1 activity? Qwen 2.5 answer. The idea that dietary components could influence the activity of specific enzymes like PSKH1, protein Serine Kinase Homolog 1, is intriguing and falls within the broader context of how diet can modulate cellular signaling pathways, gene expression, and enzyme activity. While there is currently no direct evidence specifically linking dietary components to the regulation of PSKH1 activity, several indirect mechanisms suggest that diet could potentially influence its function. Here's an overview of how dietary components might theoretically switch off or modulate PSKH1 activity. 1. Calcium regulation via diet. Mechanism. PSKH1 belongs to the calcium-dependent protein kinase (CDPK) family, meaning its activity is regulated by intracellular calcium levels. Dietary components that influence calcium homeostasis could indirectly affect PSKH1 activity. Examples. High calcium intake. Excessive dietary calcium could lead to sustained high intracellular calcium levels, potentially causing desensitization or downregulation of calcium-sensitive pathways, including those involving PSKH1. Vitamin D. Vitamin D regulates calcium absorption in the gut. Deficiency or excess of vitamin D could alter calcium signaling and thereby influence PSKH1 activity. Magnesium. Magnesium acts as a cofactor for many enzymes and also competes with calcium for binding sites. A magnesium-rich diet might reduce calcium availability, potentially dampening PSKH1 activation. 2. Phosphorylation/dephosphorylation modulation. Mechanism. The activity of kinases like PSKH1 can be regulated by phosphorylation or dephosphorylation events. Certain dietary components may influence these processes by affecting upstream signaling pathways or phosphatase activity. Examples. Polyphenol, e.g., resveratrol, curcumin. These compounds have been shown to modulate kinase activity and signaling pathways. They might inhibit upstream kinases or activate phosphatases that dephosphorylate PSKH1, thereby reducing its activity. Omega-3 fatty acids. Found in fish oil, omega-3 fatty acids have anti-inflammatory effects and can modulate signaling pathways by influencing kinase activity. They might indirectly suppress PSKH1 activity if it is involved in pro-inflammatory pathways. 3. Oxidative stress and redox signaling. Mechanism. Reactive oxygen species, ROS, and redox signaling play a role in regulating kinase activity. Dietary antioxidants or prooxidants could influence the redox state of the cell and, consequently, PSKH1 activity. Examples. Antioxidants, e.g., vitamin C, vitamin E. These can reduce oxidative stress and may prevent oxidative modifications of PSKH1 or its regulatory proteins, potentially maintaining it in an inactive state. Prooxidant foods, e.g., processed meats, fried foods. High intake of prooxidant foods could increase ROS levels, which might either activate or deactivate PSKH1 depending on its sensitivity to redox changes. 4. Inhibition via phytochemicals. Mechanism. Many plant-derived compounds, phytochemicals, are known to interact with signaling pathways and enzymes. Some phytochemicals might directly bind to PSKH1 or its regulatory domains, inhibiting its activity. Examples. Quercetin, a flavonoid found in fruits and vegetables. Quercetin has been shown to inhibit various kinases. It might similarly inhibit PSKH1 activity. Epigallocatechin gallate, EGCG, a major catechin in green tea, EGCG has been reported to modulate kinase activity and could potentially affect PSKH1. 5. Hormonal and metabolic pathways. Mechanism. Diet influences metabolic hormones, e.g., insulin, leptin, and metabolic pathways, which in turn can regulate kinase activity. PSKH1 might be part of a signaling cascade influenced by these hormones. Examples. High-fat diets. Chronic consumption of high-fat diets can lead to insulin resistance and altered signaling pathways, potentially affecting kinases like PSKH1. Fasting or caloric restriction. These conditions trigger metabolic reprogramming and may downregulate certain kinases, including PSKH1, as part of energy conservation mechanisms. 6. Gut microbiota and metabolite production. Mechanism. The gut microbiota metabolizes dietary components into bioactive compounds, e.g., short-chain fatty acids, bile acid derivatives that can influence host signaling pathways. Examples: short-chain fatty acids, SCFAs produced by microbial fermentation of dietary fiber. SCFAs like butyrate can modulate signaling pathways and gene expression, potentially affecting PSKH1 activity. Tryptophan metabolites. Gut bacteria metabolize tryptophan into compounds like indole derivatives, which can influence immune and stress signaling pathways. 7. Feedback inhibition from downstream effects. Mechanism. Dietary components might influence downstream targets of PSKH1, leading to feedback inhibition of the kinase itself. Examples: amino acid availability. Diets rich in specific amino acids might alter the abundance of substrates or products in PSKH1 regulated pathways, indirectly switching off its activity. Secondary metabolites. Compounds like sulforaphane, found in cruciferous vegetables, can induce detoxification pathways and might influence feedback loops involving PSKH1. Challenges and research gaps. While the above mechanisms provide plausible ways dietary components could influence PSKH1 activity, several challenges remain. Lack of direct evidence. There is currently no direct experimental evidence showing that specific dietary components regulate PSKH1 activity. Complexity of signaling networks. PSKH1 is embedded in complex signaling networks, and its activity is likely influenced by multiple factors simultaneously. Species-specific differences. Most research on PSKH1 has been conducted in plants, and its role in animals or humans is less well understood. Conclusion. Dietary components could theoretically influence PSKH1 activity through mechanisms such as calcium regulation, phosphorylation modulation, redox signaling, direct inhibition by phytochemicals, hormonal changes, and gut microbiota interactions. However, further research is needed to establish direct links between specific dietary components and PSKH1 regulation. If you're interested in exploring this topic further, studies using biochemical assays, knockout models, or dietary intervention experiments would be valuable to test these hypotheses.
Ask.
Explain.